sure we've got our recording going. There, you got that? Yes. So welcome to Thoughtful Thursdays. I'm Sherry Dorn, the state coordinator for Georgia's Master Gardener Extension Volunteer Program. It is my distinct pleasure to welcome Dr. Bodie Pennisi this afternoon. You all may not know this little piece of trivia, but in all of the hats that she wears, she also served as my committee chair when I finished a PhD program. Gosh, it's almost 2 years ago now, Bodie. Boy, I say that so excitedly. But she held on for that wild ride, and she's a great sport and a great fan of Georgia Master Gardeners. So we welcome her to our session today to learn about propagation of native perennials. Bodie, welcome.
Thank you, Sherry, and happy afternoon, everyone. Let's see if we're going to get some rain today or it's going to stay sunny. I think the summer is really coming on to us. It's getting to be 90s and pretty humid outside. I was there in the garden that you see on this picture yesterday and really having a blast looking at the flowers and the insects. So today for 30 minutes, I'm going to give you like the super condensed crash course on propagation of native perennials. And we're really going to be looking at only herbaceous plants. There's just not much time to get into anything else. So most of you, if not all of you, have done a lot or some propagation, and you probably have been exposed to quite a bit of information from your training days. But so some of this is going to be hopefully a refresher for you. But if you don't know something, put it in the chat and I'll be happy to answer it later. But I will go pretty fast here. So propagation, recall that we have seed propagation that we refer to as sexual means of propagation. This is where you have genes that come from the mother and genes that come from the father. And then they merge together, form an embryo that combines genetic material and information from both parents. This is where you have diversity in the population. And then you have vegetative or asexual propagation. And with the asexual propagation, what you're doing is cloning the mother plant. One plant is being cloned X number of times, and you're just reproducing the same genotype. A healthy seedling is something that we strive for every time we propagate. You put a seed, ideally you expect this seed and all the other seeds that you have spent money on or effort collecting and cleaning and storing and all that, you want it all to turn into a viable, healthy seedling. So this is a picture from some of my seedlings of putting seeds after they have been treated. We're going to talk a little bit about what kind of treatment some of those perennials require. And then if you you have done your job well, you'll turn into something as healthy as the one you see on the right. This is a picture of perennial lupine, and just a little botanical fact, because I can't prevent myself from going there. If you could see these two first pair of leaves that look quite different than the other pair of leaves, that's because they are not the true leaves, they're the seed leaves, and then you get the true pair of leaves. Just a little curio there. So type 2 seed dormancy, we have, what is dormancy? This is nature's way to make sure that the seed is not going to germinate until the conditions are totally perfect for that baby seed to start growing. So we recognize primary and secondary type of dormancies, and those dormancies can be quite complex. And with perennials plants, even some annuals, you almost always have some kind of dormancy. With shrubs and trees, almost always have some kind of dormancy. So this is a pretty complex field, and I'm going to try to make it as simple as possible, but so that you understand a little bit of the physiology behind it, because you are scientists, you have been exposed to a lot of scientific information, so you're very knowledgeable, and this hopefully will come as more interesting information, and you can read on your own a little bit more in detail. But we recognize the primary dormancy that exists with the seed when it is mature. And so when the seed is newly dispersed, it fails to germinate under normal conditions. That is when the seed exhibits a primary dormancy. We call this primary dormancy. Secondary dormancy is the one that you're most familiar with. This is after you have completed those conditions that have been met to delay germination and the primary dormancy has been satisfied, the seed still is not going to germinate. And that is because you actually have secondary dormancy. And that secondary dormancy is really what we're going to be talking about today. The secondary dormancy can come from things like hard seed coat, things that, you know, I mentioned that I'll be talking about herbaceous plants, but this is a good example of a plant that has primary and a secondary coat dormancy that is mechanical, that is coming from the hard seed coat. So we do something called scarification to soften and weaken those seed coverings, and that's an eastern redbud. And I'll be showing you some of the lupine, which are pretty much quite a few of the beans have really hard seed coat, and that hard seed coat almost always requires some treatment in order to weaken it and make sure that there's a crack to which the water can get through. Then there's another kind of dormancy that is chemical. We also call that inhibitor dormancy. This comes when you have a fleshy fruit that will not separate from the seed easily. So nature's way of keeping that seed from germinating is to enclose it in something that is going to keep it from germinating. And so germination inhibitors naturally exist in the fruit pulp. And here in this picture, a series of three pictures of chokecherry, where you could see how the fruit comes fresh from the tree, and then you make sure that you grind it up in a large batch. Depends on how many seeds you're trying to process, and then clean up that pulp, and then you're going to end up with the seed. So this is actually, the last picture is a false Solomon seal, and it's a very similar way of cleaning the seed, removing these fleshy coverings, and leaching in running water for 12 to 24 hours. That's important to have a running water because these chemicals are water soluble, and so you have to keep leaching so that they dissolve in the water, and then therefore they're removed from the plant, from the seed. Stratification is what we call a collection of techniques that provide the seed that has imbibed water. In other words, it's already at that stage where it has all the water it needs. And then this one is going to have moisture and air and temperature that is appropriate for that kind of dormancy that is overcoming, and then the time that it requires for that dormancy to go through. So the definition is to layer seeds in moist medium and to place it in cold 35 to 45 Fahrenheit, or warm conditions 68 to 86 condition temperature for about 3 weeks plus. And that 3 weeks plus can vary to almost 3 months. And that is just the way it is. Looking at the plant, you'll never be able to tell this. This is all from research that people have spent years and years compiling. The environmental conditions, and most of the time you're going to be doing moist chilling. So you always have to put in water first. You have to moisten the seed in order for it to be brought to that water status, moisture status, to initiate germination, and then come in the chilling, the temperature, and for a certain length of time. So that's moist chilling. So imbibe the seed to at least 50%. Make sure there's enough air, because all the processes that are going to be starting are intensive and requiring respiration. And if you recall from your training days, respiration is the process by which plants obtain energy after they have fixed carbon from the air. They have all the starch resources. Now they're going to have to start breaking those, whether proteins or starch, and then utilizing the energy that they stored. So respiration needs oxygen. Oxygen is where that need comes from. Temperature has to be higher than 45 degrees. That will slow after ripening. And the time, 1 to 4 months for most temperate woodies in this case. I'm just giving you a little bit of a span there. But again, it can be a pretty good long time depending on the species. So what happens behind the scenes, so to speak, when this stratification is taking place? Well, basically you have an undeveloped embryo. It's like a preemie baby. That preemie baby is going to be placed in a perfect condition after it's been separated from the mother plant, and it is going to ripen. It is going to complete its development and be ready to mature. And in this series of pictures here from my plant physiology text I like to use in my class is showing you a carrot, either carrot or magnolia. I can't tell you which one it is because I can't remember. They could be either one because they both require similar conditions. And what you're looking at is the seed at the beginning of 12 hours. It's how little it is. Let me just grab some pointer here so that I could show you. So there is the embryo, and this is the entire seed. So the seed coat is the darker covering on the outside, the outer line, and then inside is the endosperm, which is the nutritional tissue that the mother plant has put aside for that embryo to use when it's germinating. So when you have imbibition of water and then you subject it to cold temperatures, you notice how this is doubled in size in 6 hours additionally. And then some of that is starting to liquefy and be used in that development of that embryo. Not a whole lot. It's going to be used mostly later, but some of it is used in this time as well. At 30 hours, more double the time, then you could see that this is starting to really shape into more of a what we call an axial look, where you have clearly radical, or that root that is pointing downwards, and clearly leaves that are developing as a shoot. And then at 40 hours, you can really see a lot more. So if you look at that, it's about maybe 5 times larger. So this is what's happening behind the scenes, if you will. That is the preemie baby getting into full term and then be ready to go and make sure that it's germinating properly. So how do you do this? Simply cool, moist stratification. My refrigerator is full of little jars, little baggies full of sphagnum moss, sand. I use all sorts of media, or just plain paper, moist paper towels. Some of them don't have labels on them. Who knows how long they've been there? I learned my lesson a long time ago. I started throwing away a whole bunch of them. But it's the best thing to do is to make sure that you find the smallest amount of space and whatever you need in order to recognize what the information is. When did you put it in? What the species it is? As much as you can in there. I usually just keep my bags from the seeds. If I have purchased those, I would keep them either stapled to that or in some other form and fashion so that they are right there. And I always snap a picture with my phone so that I can go back and say, okay, that's what I did on that date, because you can get 100 of them in there. And if you have more space or a dedicated space for this, if you're making, for a friend, or you're working for a garden or something and you need more, then you can definitely invest in something a little bit more larger, and depending on how you're putting them in, if you have some larger material, you can then spread it out. But this just gives you just a refrigerator is all you need for this. Alternatively, put it in soil and over the winter. That's exactly what happens in nature. After ripening is something that happens with not so many species, but there are some that really require this. And again, you would not know it. It's like a secondary dormancy that is imposed on the plant. It's like the first conditions are met, the chilling, the length of it, and then the plant requires now warm condition for some reason. It needs a little bit more. So it's really reluctant to start going. So immature embryos require a warm period to continue that development. And then after that, also another cold period to break the dormancy. So you can have 1, 2, 3 of those following: cold, warm, followed by cold. So hollies, plums, ironwood, lilies, bluebells, bloodroot. In here, there's a picture of bloodroot, and a trillium are in that category. And there is Ilex verticillata winter red here, which is telling you that first it was in the warm for 30 days, then it was cold 90 days, and there were 134 seeds in there. So moist sphagnum moss is good because it helps prevent mold and mildew from growing on slow to germinate seeds, because in that period of time you may have some of those conditions that will be favorable for undesirable kind of organisms. This is from a picture from a place I like to buy my seed from, Prairie Moon Nursery. I'll mention to you some sources of for native plant seeds. And I just wanted you to see that once you get those, you will see some of those have a tag information that says refrigerated seed. So this seed has been stored on the refrigerator until shipped. Keep in the refrigerator until planting or start pre-germination seed treatments. So make sure that you pay attention to these. There are several that may come, and that will be very important. On the tag, on the well-packaged place, will give you things like germination code. It tells you exactly what A, B, C, D, E, L, that particular company keeps track of these. So in order to germinate, this particular species of wild ginger needs more warm, moist period followed by cold, moist periods. And so again, just make sure you pay attention to these and give them the best that you can for the length of time, and you'll have much higher success. And even with that, sometimes the tag will say 50 or 60% germination expected. It just means that naturally that germination is low, even though you may have met all the requirements. So nothing on you, it's just the way it is. For methods of scarification, if you are going to be breaking through the seed coat or scarifying it, making a scar, nicking it, doing anything, if you're having a few large seeds, easy enough to do. I'll show you some pictures. But if you have large amount of seeds, large batches, you can do hot water scarification, which is just dropping the seeds into 4 to 5 times the volumes of hot water, not boiling, boiling water, but then you remove it and you don't cook the seed, and allow the seeds to soak and cool for 12 to 24 hours. After that period of time, they need to be planted. You cannot just sit around and wait for this or put it back to dry, because you have already broken through that seed coat and water has gotten inside there. Acid scarification is not something I recommend to most of you, but again, it is something that is very effective, and it certainly is standard practice in large batches of seeds processing for nurseries and places like floriculture operations. So these seeds are placed in sulfuric acid, and after several, depending on what kind of plant you're looking at, whether it's a thick seed or a little bit thinner seed, determines how much time you put it in. There are a few minutes to say like 10 or 15 minutes, sometimes, an hour, depends on the thickness. And then you always rinse those in running water, not just sitting water, but running water. You want to make sure that there is no acid that is left. And then you can also do mechanical scarification. And let's take a look at some of those examples. If you have, say, Bacteria australis, which is false indigo, a gorgeous, gorgeous plant, or something like a spurred butterfly pea, they are both in the bean family, and they both need someone to break that mechanical dormancy imposed by the hard seed coat. So there is the seed, and it's pretty hard seed. So what you're going to do is you're going to either warm soak them, or if that didn't work, then you can just go ahead and do something like scratching, nicking, or using sandpaper, anything to get through that seed coat. And you have to be pretty careful how you are touching those small seeds, especially if you have some kind of like a razor blade, like the picture shows you. I'll show you some examples, but that's about the right size. I like to take 2 pieces of sandpaper, large coarse sandpaper, and then just rub them gently in a single layer, back and forth, back and forth, back and forth. And that is usually enough. And that tells you that the radicle, which is the embryonic root, is starting to emerge after 48 hours soak in water. If you do get these conditions and that starts happening, then you're in good shape. But again, if it doesn't work, then or you want to try just scarification to just compare them and see which one works best, you can go ahead and do that. Seed scarification by nicking, breaking, softening, or otherwise weakening the seed coat to speed germination is the term we use, seed scarification. Again, sandpaper smaller seeds. If you want to grab the seed and just run it on a nail, or nick it with some nail clippers, or anything that, again, just creates a little nick in there so that the inner coat, something a little bit is seeing. This would be a little bit too much already. This is already going into the embryo, so that's a little bit too far, but something like this is perfectly fine. And some seeds, believe it or not, are incredibly, incredibly tough. Photodormancy is another kind of dormancy that we see primarily with herbaceous plants, and lettuce is a perfect example, and so is butterfly weed, which you see a picture right here. Butterfly weed is a milkweed species. I'll show you some examples of propagation. And so what we require is for the seed, in order to germinate, it needs to be on the soil surface to see light. It will not grow in the dark like most species. And some showing you here how I clean my milkweed from the fluff that comes with the seed. I just put it in a portion of a heavy stocking, and then just rub it in between my fingers, and seeds come from one end, and then the clean seeds come out from the bottom. And I always look through to see which are the small seeds that look like they don't have a large viable embryo. Not necessarily, it's not always the case that smaller seeds will not produce embryos, but I'm just saying, to be on the safe side, if you have plenty, just go through and pick out the largest, plumpest seeds and discard the chaff. And this way you can also store them too. Here's a picture of seeds that I have put circumventing the whole light propagation, just putting under mist, because I do have the capability, and that's easy enough for me to do. Just put it under mist, and they germinate actually even better than if you just leave them on the surface of the soil. And even breaking the code that is cold stratification for 30 days. If you keep it under mist, but it has to be a really consistent mist day after day, you can break dormancy that way. So anyhow, this is without any kind of stratification on the seed. This is just mist. So just give you an example. And this is what these seeds look like once they start growing. Again, it's amazing to see how early that tuberosa root develops. That is the species of milkweed. It means that it has a tuberous root. And you can see that this is developing very, very early. Even half that size on the seedling, you can see little tuber starting to develop there. What do you do with things like tiny seeds, which say the lobelia, which is a great, great plant, but all the lobelias have that very tiny, tiny seeds. And you can see the picture here. I mean, honestly, you can barely see these dark little specks against pure white craft sand. So what I do is I mix it with that white sand, and it's cleaned from the craft store. It's very small, very grainy seeds, uniform. And then I just, shake it pretty good. This one bag can give me a whole lot. So honestly, lobelia is like one of the easiest plants to propagate, and it gives you almost 100% every single seed packed is germinating. So you mix it in, and then you put it on the surface of the soil. And I use light colored sand. White is really ideal for this. And this way I can actually see where the seeds went. So that's a helpful little tip, I hope. Vegetative propagation, how are we doing on time? I'm a little bit over time, so I'm going to speed up even more. So the vegetative propagation is really, really cool. For years, I was just into vegetative propagation. I was like, nah, I'm not going to bother with seed. It takes too long. But cuttings is certainly something I knew quite a bit. But nowadays, I do both equally. The goal in propagating by cuttings is to replace the missing part, whether it's a root or a shoot. If it's a root cutting, then you replace the shoot. If it's a shoot cutting, then you need to grow roots to replace what was lost. The thing about cuttings is when you're out and you're taking those cuttings from a place, whether it's a friend's yard or you're somewhere out in the wilderness and you're allowed to collect cuttings, I always carry with me a little pack of cold and moist, so a paper towel or two that have been pre-moistened in a plastic bag with some cooling agent, something to keep the cold, and in a little ice bag, and I pack it on my backpack with some clippers. Don't forget the clippers. So wherever you're going to be taking cuttings, and those cuttings can be pretty long shoots. This happens to be mint here, and I didn't have that nice bag there. I just used some garbage bags I had around from the grocery store. Sometimes I've used some other things too, but important is to keep them moist and to keep them cool. That keeps the water status of these actively growing tissues from losing moisture. And then you can stick them in the refrigerator when you have time to work on them. I mean, they can be up there for a week. I've kept things that are more succulent even 2 weeks with no problem. Another really easy way to propagate with, especially when you're working with something that is producing that crown that has lots of roots and growing points, like the false sneezeweed you see here, we are seeing easily split apart parts of the system that has a shoot, like this one right here, and some roots already on it. So something like this split from the crown, which is right here, from one plant, I easily got about 30 separate, and I could have easily gotten more. So easy to do. Divisions work for many native perennials. Another example is Penstemon digitalis, or the eastern bird tongue. I love that plant. I love all the Penstemons, but this one is one of my favorites because it gives you a little bit of a larger bloom. It's not native to Georgia necessarily, but it's, we can grow it, especially more in the northern part of the state and in a little bit more cooler gardens. but very easy to propagate, very, very dependable from seed as well. But once you do get them large, then you're kind of like, why do I have to wait for the seed? So you just go through a nice clump and you start looking at anything that separates a single shoot. So there are 2 shoots right here in this part. There are probably 2 to 3 even here, but any one of these with a little bit of root on it or no root. I don't usually use rooting hormone if you were to ask me this question on perennials. If there is any kind of woody tissue on it, then I go ahead and use about 1,000 parts per million. That works really well. But you can divide a large clump into many small ones, or you can say 1 clump and divide it in 2 or 4, however, how many plants you want to, and as quickly you want that plant to turn into bloom, because something like this is going to take 2 years to form blooms, right? Because it needs to bulk up first before it starts putting out flowers. If you start with something large that is easily about, say like, 6-inch pot, and you split it in half, chances are you're going to have them blooming the next year. And once you do that, when there are no splits that have roots on them, it's always a good idea to keep them in the shade and water them well. Again, I have the ability to put it under mist, or at least keep it in a part of the greenhouse that is cooler and shadier, and that keeps that loss of moisture from the leaves until they start forming roots, which they dependably do. At this point, you're far more better off keeping them a little bit cooler and not overwatering them than overwatering them and keeping them under sun or sunnier conditions. Cooler and shadier is really what they need at this point. How do you achieve the best form in many of these plants? This is an example of a very easy to propagate blue flax, Lineum perenni, which again, it starts growing and it starts forming this spindly little unhappy looking at some first glance form. Well, what I do is once I have them growing well, then I go ahead and I give them a good haircut. And we're talking about taking like 2/3 of that. And very quickly it starts bulking up and becoming stronger, and also putting lots and lots of breaks. And so that creates a form that is stronger and also makes sure that you have multiple shoots to produce blooms. So that's the other benefit. And you can do that with practically anything and everything. This is New England aster. And again, pruning it literally or clipping, depending on the size. I mean, you can do them when they're smaller, you can do them when they're a little bit older. This is an example of when they're a lot older here. Again, this is from a single year. When you split, or it is, excuse me, when you clip or prune the main tip, the auxin kicks in, is removed, and then you release some of the buds from underneath to start breaking, and then you have this branching going on. And then from 1 shoot, now you have 2 shoots, and you can have even 4 or 5, and then forms that nice bushy plant with the bloom on. Propagation media. Next question is, what kind of media can I use to propagate? Well, practically anything that provides air and water relationship of 25 to about 40% airspace. So as long as it's clean and as long as it's aerated, you're good to go. So for most plants, 2 parts coarse perlite to 1 part peat by volume, and it works really, really well. Important thing to consider is when you're starting, whether you're growing seedlings for vegetables or propagating native perennials from seed or anything really, propagation during the unfavorable time of the year, which is the winter, is going to benefit from some kind of root zone heating we call bottom heat, and that needs to be about 70 degrees Fahrenheit. And you're far better off if you're like in a greenhouse. We know that there's no sense of pumping heat into the whole greenhouse. You put it where it counts, which is where the root system is. And so these are, believe it or not, this is heating element underneath the benches here, but for us, we're looking at something like heating mats that we can put on the bench, or we can put on a growing stack, or whatever setup we have at home. And important thing is the heat is where it needs to be to speed up the process. Okay, native seed sources. You can look at the Roundstone Nursery, the Prairie Moon Nursery. American Meadows is another one I forgot to put in here, but also State Botanical Garden of Georgia. And many botanical gardens will mail you packages and will even sell seeds. If you want to go and collect certain ones, or talk to someone that is working with the plants, they'll probably be more than happy to share with you some seeds. So that's where I get a lot of mine. And with that, thank you so much. And I'll just keep on going. Thank you very much. And Sherry can take it from here.
Woo, Bodie. She did it. She did it in like 29 minutes, 29 and a half minutes. Thank you so much. That's fascinating. Yeah, so any questions from you all? It's right at 2:30, so we'll take a couple. Sue says, what is the name of the nursery? Not on the screen, the one that you,
American Meadows.
American Meadows. Okay. All right. Bonnie, I see your hand up, the raised hand icon. Are you asking a question? If so, you can go ahead and unmute yourself.
Yes, my question was, I finally got it typed in. I have quite a few penstemons and they are producing seed. Is that seed viable if I collect it? Or am I
That's a really
better-
good question. And believe it or not, this morning going through my greenhouse, I was asking myself that same exact question because I do have some penstemon. And I'm like, are you going to be producing that seed? And I'm like, okay. I think I'm going to go ahead and try to propagate from seed. Once I feel like it's completely dry, I will remove it and then I'll clean
Okay.
some of the material, and I'll probably just store it up to maybe about, a couple of weeks. And then I will go ahead and give it good. If I'm remembering, 30 days of cold stratification was sufficient, but just to be on the safe side, if you want to split in 30 and 60, just to be safe, with 60 days of cold stratification, you should be pretty safe. But if you look up online and it says 30 days is enough for that species, then no reason to go with 60 days.
Okay.
And just do the same thing. And yeah, it's penstemon is one that also has very, very tiny seeds. Yeah.
Okay, okay, thank you.
All right, we've got a question about softwood versus hardwood cuttings.
Softwood versus hardwood cuttings. Certain species propagate very easily from hardwood and some others do not propagate from hardwood. Again, I didn't have time. It was not about woody plants, but to answer your question, it depends on what are you really trying to do. I mean, I understand you're trying to propagate, but the reason why I'm asking is if you're in the nursery business, you are meeting different kind of deadlines. And so if you only can take cuttings from, say, July through beginning of August, or, end of June, and that's all you can do, then this is what you try to propagate. But if you have the ability to take over a longer period of time and you're just trying to learn something, you can be taking semi-hardwood cuttings, hardwood cuttings, softwood cuttings, another thing, because some plants will propagate very easily from softwood cuttings, only it has a narrow window that you can collect that. And you also have to be very, very careful and cognizant of all the needs that need to be met. So mist is a must with softwood cuttings. But they propagate a lot easier because they don't have that much, wood on them. So there is no preferred method. Some plants are easier to propagate from semi-hardwood cuttings, others from softwood cuttings. And the hardwood cuttings is there for people that take it in the time that is less busy. So for example, if it takes like a whole 6 months for something like a grapevine to form roots, well, they just stick it out in the field and they wait for it to form roots, which is again fine, but that's something that is meeting the needs of the nursery industry and not necessarily the homeowner. I hope I answered your question. But materials, Durr's book is like the standard text for looking at what kind to propagate and for how long. And of course, time. Some things propagates really fast, sometimes a little bit longer. Those are the questions you need to ask yourself.
So a request for a recommendation for additional information, the best online resources.
Oh, well, Sherry, you and I need to finish that, that publication. Online resources, I still go back to Durr's book on woody plants, woody plant propagation, whenever I really want to go to someone that I trust, resource that has been compiled over years and years of work and research. When I look at online stuff, I mean, I go to places like the Prairie Moon Nursery, the Roundstone, and others. They will have quite a bit of information on where you can read up on even some, how do you collect seed? How do you store the seed? So if a place has a lot of information, I tend to go and work with them. I even send them questions, and most of the time they're very happy to answer me back. North Carolina, floriculture, and I think it's actually a master gardener. I'm trying to think of it, Sherry. I think I've seen some really, really good databases coming out of them that have information. I just can't think of one right now that has like a comprehensive list of species with the plant, with how do you propagate them.
Yeah.
Do you have any idea? Do you know any?
I don't. That's rather proprietary information. I would look. I keep expecting the State Botanical Garden, truthfully, to come out with a book or a publication or something like that.
There is a publication by the State Botanical Garden on native perennials and propagation. There are 30 native perennials with pictures, and so it's a free. You can print it. It's on the State Botanical Garden of Georgia website. So if you haven't seen it, I would urge you to go ahead and check it out.
And of course, if we're looking for information on natives, then we have the whole series, the whole publication series available through the UGA Extension publication website.
Right.
Yeah. And then how can I make a high humidity environment so I don't have a misting system? Boy, y'all, I can't wait to share with you the latest chapter in our handbook revision. We've got all this kind of stuff covered in color photographs. It's coming, but Bodie.
There are quite a few tricks you can do. Again, the length of time that you need to keep things moist and how often you're going to be able to check on them is really important. You can get away with certain species with less misting if the leaf is a little bit harder, if it has a thick cuticle. But if it's something that is very, very soft and it loses moisture, and you're working full time, it may be that you're going to have to invest in something a little bit more advanced and pay attention to where you're putting those plants. I have propagated at home without any misting system with just a sheet of paper of plastic above a tray or a pot, and literally just misting. But it takes longer because you don't have the ideal conditions, but at home at least the temperature is more permissive. So it's about how patient you want to be and how much you're willing to experiment. So start with something that is literally foolproof, that is going to form roots really quickly. Let's say like coleus. I mean, it forms roots in water. But I'm just saying, get used to a little bit by little bit, maybe moving it not in water, but in soil, mist it, watch how quickly the mist disappears, and go from there.
Yeah. All right. Well, that wraps up our questions in the box. Thank you. I have a number of questions myself that I'll bend your ear about the next time you pass through my building.
Sure.
So a few announcements for all of you all. Just a reminder that Thoughtful Thursdays does continue. This series does continue through November. So our next meeting time is August 12th at 2:00. We do record our sessions, and those recordings are available within a few days within your MGLog account. So you can log into MGLog, and actually, Bodie, if you can advance to the next slide. That image there on the screen is in the bottom left-hand corner of your landing page when you log into MGLog, and you can click at that View All button and see all of our archives. And this is our 4th year of Thoughtful Thursdays, so there's quite a few archives there that you can catch. And don't forget to record this as continuing education. You get a half an hour of continuing education each month. So next slide, please. Just a note about RMT. Many of you have been asking, because July 1 is our magic number. We are still waiting on our update from our 4-H program area. So this is a little bit later than we have anticipated. So just watch our messages on Trellis and coming through email and from your extension agent. And as soon as that is available to us, we will let you know so we can go ahead and get that updated. All right, next slide, please. And as I mentioned earlier, this is actually a page, maybe, no, it's not. I started to say this is a page out of the propagation chapter, but it's not. We have been working, y'all, for 2 years on this huge overhaul of our Master Gardener Handbook, and we are so excited. We are so close that Sarah and I can see the end in sight, truly. And we will be having some details as soon as I confirm our prices with our printer. So we'll be having some of that information available to you. If you haven't bought a handbook in a while, you know, maybe if you trained a few years ago, you're really going to want a copy of this book. It is full color. It is exciting, new chapters, new content information, and the plant propagation chapter was totally overhauled. So if propagation is your thing, you're going to love, love, love that new chapter. So just a little plug, keep your eye out for promotional information. We'll have it on Trellis. We will have it, you know, sent to you by email. We're going to let our agents know when that's ready to go. So just keep your eyes open and your ears open, and you'll be able to get that information for a new copy. All right, so I think there might be one more slide there. Yep, so our next upcoming Thoughtful Thursdays, like I said, we meet again August. Becky Griffin will be with us. She'll be talking to us about seed saving, which I know is a popular, interesting topic as well. And also I bet she'll be telling us a little bit about the pollinator census, because that'll be coming up shortly after our Thoughtful Thursdays. In September, we'll hear from Dr. Waltz again, because he gives us our fall turf grass update, what to expect and what to look for. And then we'll have a session in October and November. And I got to get those topics put up here for us all. So glad to have you with us, and we will be seeing you again in August. And Bodie, thank you so much for being online with us this afternoon.
Good seeing y'all. Have fun.
All right.